Okay, so hopefully this one will run a little bit smoother. The next part of this presentation is going to be looking at immunity. Specifically, we're going to focus on innate immunity. So our immune system is how we defend against different pathogens like viruses, bacteria, fungi, and other microbes that can cause disease, and then also some non-living disease agents. So how we combat poison ivy toxins or other allergy-producing chemicals, maybe from food or cosmetics, or sometimes latex gloves. And we have two broad forms of defense mechanisms. Like I said, today we're going to be focusing on innate immunity, so those are the defenses that we're born with. And then adaptive immunity are the mechanisms that we develop over time that defeat a pathogen and then leave a memory so that we can defeat that pathogen again quickly upon any future encounters. So the way that innate immunity is distinguishable from adaptive immunity is that it has a more local effect as opposed to a systemic effect. So an example of this would be like if you get a mosquito bite, right? You get a little bit of inflammation in that local area, but very little effect elsewhere. The one exception to this is that fever is part of your innate immune response, and that does have a systemic effect. Innate immunity is also nonspecific, so that means it acts against a broad spectrum of disease agents. It doesn't target any one particular pathogen or work against any one particular pathogen. And finally, it lacks memory. So next time you encounter a pathogen, your innate immunity has no easier time, defeating that pathogen than it did the first time it was exposed to it. So there are three types of defenses that are employed by innate immunity. We have protective proteins, which we'll talk about as we move through, protective cells, which we've already talked about a little bit if you've watched the PowerPoint on the lymphatic system, and then protective processes like fever and inflammation. So there are three lines of immune defense, and the first two are our innate immunity. So the first line of defense is our epithelial barriers. That's going to be our skin and our mucous membranes. And so those are really impenetrable to most of the pathogens. Then the second line of defense is the line of defense that basically kicks in if those pathogens are able to break through those epithelial barriers. So these are going to be our cells like leukocytes, macrophages, which we already talked a lot about, those natural killer cells. Also antimicrobial proteins, which we'll learn about in this presentation, and then fever and inflammation are two protective processes that our immune defense triggers to beat pathogens. And then the third line of defense is adaptive immunity, which we'll talk about next time. And those are mechanisms that help us to defeat a pathogen and then leave a memory so that next time we encounter that pathogen, we can beat it even more quickly. So we're going to talk about our first line of immune defense, and that's the epithelial barriers. So some of the important aspects of epithelial barriers that help us in terms of our immunity are the antimicrobial aspects of skin in general. So we have sweat that contains antibacterial components like lysozymes. We've talked about lysozymes a lot. There's also cells like keratinocytes, neutrophils, which are white blood cells, and macrophages that live within our skin. And those guys actually produce proteins. And those proteins are important because they destroy different bacteria, fungi, viruses, and other pathogens like that. Mucus is another important aspect of our epithelial barriers. So some of our organ systems are actually exposed to the external environment. And some examples of that would be like your respiratory tract, right? Your reproductive and urinary and digestive tracts, they're all exposed to the external environment. And so they have to be protected by mucous membranes. And mucous membranes contain a sticky substance called mucus. And mucus is really important because it traps pathogens. So, for example, if you were to inhale a pathogen, then that pathogen would get trapped in the mucus, within the mucous membranes of your respiratory tract. That would be moved to the nasopharynx by your cilia, and then you could swallow the pathogen and it would be destroyed by your stomach acid. Mucus is also important because it contains lysozymes just like tears and earwax and sweat. Hyaluronic acid is a substance that we find beneath the tissues of the skin and the mucous membranes, and basically it gives that layer a very viscous consistency, and so that makes it really difficult for different pathogens or microbes to penetrate or migrate through that viscous layer. So that's the epithelial barriers of the skin. That's our first line of immune defense. The second and third line of immune defense consists of a lot of things, but our immune cells are part of that. So the leukocytes, or our white blood cells, are neutrophils, eosinophils, basophils, lymphocytes, and monocytes. So hopefully you remember that from when we talked about white blood cells. The lymphocytes consist of T cells, B cells, and then natural killer cells, which we just talked about when we talked about the lymphatic system. And then macrophages, we have down here, are our phagocytic cells that attack microbes, okay? So when the microbes get through the epithelial barrier, or that first line of defense, we have these macrophages or phagocytic cells that will attack those microbes and engulf them. Okay, so the first of the immune cells that we're going to talk about are neutrophils. And so neutrophils are white blood cells. We talked about them a few weeks ago, and their job is to engulf microbes. Okay, so they can destroy microbes by acting as phagocytic cells or macrophages, but they also can kill bacteria through something called degranulation. And if you remember back probably the week before spring break, we talked a little bit about degranulation, but basically what happens is they have all of these enzymes floating around in their fluid, and when they degranulate, they push the enzymes to the surface of the cell to be expelled into the extracellular fluid. And so that by expelling these enzymes, they catalyze a reaction called respiratory burst. And basically it's just the production of these three chemicals down here that are really toxic to bacteria. It's superoxide, hydrogen peroxide, and hypochlorite. And so that's going to kill all of the surrounding bacteria, but unfortunately it also kills the neutrophil itself. So the neutrophil basically sacrifices itself, but in doing this is able to neutralize a lot of pathogens at once, or a lot of bacteria at once. Okay, so again, degranulation, that's pushing these enzymes outside of the body, or outside of the cell body, and then from there it's going to catalyze the reaction of the respiratory burst, which is just the reaction which produces these three chemicals that are really toxic to bacteria, but also unfortunately toxic to the neutrophil itself. Eosinophils are another type of white blood cell that hopefully you guys remember. They are big in protection against parasites and allergens. So we talked about this when we reviewed white blood cells, but they kill tapeworms and roundworms, and they also use sort of the same methods with superoxide, hydrogen peroxide, and other chemicals or proteins that are potentially toxic to these parasites. And they also have the ability to phagocytize or engulf antibody-antigen complexes. So if you remember, we talked about antigens and how our body uses antigens as a means to recognize, are these tissues my own or are they from a foreign material? And so what happens is when your body recognizes antigens that are from a foreign body, then these antibodies are going to bind to the antigens and basically flag them and say, hey, these are not supposed to be here. So then your body will respond by sending phagocytic cells to destroy these cells that have been marked by antibody-antigen complexes for destruction. Okay, so eosinophils are one of those cells that phagocytize these cells that have been marked for destruction. Basophils are another type of white blood cell, and they're really important because they secrete a whole bunch of factors that are going to activate or attract or enhance the function of other white blood cells. So these guys secrete factors that attract other leukocytes like neutrophils and eosinophils. So let's say you have an injury or an infection. these basophils will secrete factors that'll say attract the neutrophils and eosinophils or other leukocytes to that site. Basophils also secrete histamine, which is really important in dilating your blood vessels. Okay, and that's important because when we dilate blood vessels, we increase blood flow, which is really important for inhibiting infection because it's going to deliver the leukocytes more rapidly to whatever area we need them to get to so that we can remain protected from infection. Basophils also secrete something called heparin. So, for any of you that have veterinary experience or medical experience, you may be familiar with heparin as an anticoagulant. In green top blood tubes, we put heparin if we don't want the blood to clot. And so, what the heparin does is it stops the blood from clotting in these areas where we have infection so that the leukocytes so we can increase blood flow, just like the histamine, just in a different way. So, we can increase blood flow so that we get those leukocytes to wherever they need to go. They're able to mobilize more easily so that they can protect us from infection. Lymphocytes are another type of white blood cell that we talked about. So we talked about all of these three types when we reviewed the lymphatic system. Natural killer cells function in innate immunity. So we talked about those already. They're the ones that attack foreign bodies by binding to them, and then they release perforins, which then release granzymes. So the perforins poke a hole in the cell, and then the granzymes destroy all the enzymes within the cell so that the cell dies and will be consumed by a macrophage. So these guys are going to target bacteria, transplanted tissues, and host cells, which again are just cells of your own body that have been maybe infected by a virus or cancer. B cells and T cells both function in adaptive immunity. So we're not going to focus too much on these today because right now we're going to talk about innate immunity, but these are the cells that are involved in defeating a pathogen but then leaving a memory. Okay, so next time you encounter this pathogen, your body has the antibodies to fight it. Monocytes are another type of white blood cell that we talked about when we were learning about leukocytes. So they migrate from the blood and they enter into connective tissue, and once they get there, they turn into macrophages, which again are just phagocytic cells. So they're going to engulf things like tissue debris, dead neutrophils, bacteria, and other types of foreign matter. Okay, so those are all of the second line of defense in terms of immune cells. So if we go back to this slide here, see second line of defense, leukocytes and macrophages. That's what we just talked about. Now we're going to start talking about, and we actually talked about natural killer cells too. Now we're going to start talking about antimicrobial proteins and what role they play in the second line of immune defense. So the first of the proteins that I want you guys to know is interferons. Okay, and these guys are secreted by cells that have been infected with a virus. So if you look at this picture over here, you can see this cell is being targeted by all of these little viruses. Okay, so the virus has entered the cell, and these interferons are being released. They're not really going to do anything for this cell that's already been attacked in particular, but what they do is they target all the neighboring cells. Okay, so they're going to bind to the neighboring cells. And it's basically just a way for this cell here to alert all of its neighbors and say, hey, I'm being invaded by a virus. You guys should take whatever action is necessary to protect yourselves. And so what the neighboring cells do in response to that is they activate systems that synthesize proteins that will break down these viruses. Okay, so now they're more prepared for an attack, and that prevents the virus from being able to replicate itself. These interferons also target natural killer cells and macrophages so that they are ready to come engulf and destroy any cells like this one that's been infected or is dead. And then the other proteins that we're going to talk about are part of something called the complement system. So the complement system is a group of 30 proteins that are synthesized by the liver, and they're activated when they are surrounded by pathogens. So if you guys remember when we talked about the blood clotting cascade, and I showed you there's all these big reactions, this is very similar to that. I don't really care that you guys remember this big long cascade, but what I do want you to take away from this slide is that the result of that big long cascade of reactions, so one protein is developed and it catalyzes the reaction of another and another and another, and basically the end result is we get inflammation, immune clearance, phagocytosis, and cytolysis. So those are the 4 methods of pathogen destruction that are the result of this big long cascade. So the first that we'll talk about is inflammation, and this is one of the protective processes of the immune system. So if you're trying to kind of organize your thoughts, let's go back here. We talked about the first line of defense, the second line of defense, and so we've talked about leukocytes, we've talked about macrophages, we've talked about natural killer cells, and now we're talking about antimicrobial proteins, which this complement system is made up of all of these proteins that have antimicrobial functions, and inflammation is one of the processes that's basically catalyzed through these antimicrobial proteins. Okay, it's part of your second line of defense, or part of your innate immunity. So let's go back here. Okay, so there's 4 cardinal signs of inflammation. That's redness, swelling, heat, and pain. And inflammation is indicated by the ending itis. Okay, so there's all different types of inflammation. We could have arthritis, we could have encephalitis, we could have dermatitis, and so we just use dermatitis here as an example. But it is something that could be the result of a mosquito bite, it could be the result of poison ivy, or a sunburn, or blisters, maybe from running shoes or something like that. And so there are 3 major processes of inflammation, and we're going to talk about each of them individually. So the first process is mobilization of the defenses. So this is the first process of inflammation. And so we need to mobilize all of these different parts that are going to help with our immune defense. And so we do that through something called hyperemia, which is just increased local blood flow to the tissues where it's necessary in order to quickly deliver these white blood cells. And so also what it does is we increase blood flow with this hyperemia, and that allows us to wash away any of the toxins or the metabolic wastes from these tissues more rapidly. Okay, so this increased blood flow is what gives us the heat and redness that we see with inflammation. The next important factor in mobilization and defense is selectins. So these are molecules that adhere to the surface of the endothelial cells. So here in this picture, let me just break this down for you. We have let's just say a blood vessel here, and then we have this site of inflammation here because we have a splinter. And so what happens is these selectins are going to line, these molecules called selectins, which I don't think are labeled in this picture, but you can imagine them. They basically line the endothelial cells here. And when they do that, it makes the endothelial cells sticky. Okay, so that's really important because when they're sticky, the leukocytes, in this case neutrophils, are going to move through and stick to the surface of the endothelial cell where we have damage, or where we need where we need the leukocytes to basically take action. And so when that happens, it slows down the leukocytes, and they're able to cross through in between these gaps between the endothelial cells. And that is called diapetasis. Okay, so that's what we see here. So selectins cause the leukocytes to stick to the endothelial cells so they can slow down and crawl through basically these endothelial gaps. So that's called diapetasis. And then they enter into the tissue fluid of this damaged tissue. And then finally, the last process of inflammation is containment and destruction of pathogens. So we don't want these pathogens circulating. We want to keep them in a small area and to destroy them. So the areas surrounding an injured tissue are going to clot. Okay, so we get a scab up here, and then maybe we get a scab down here, or clotting down here. But in here, the tissues or the cells are going to secrete heparin. And so we already know heparin is an anticoagulant, and it's going to keep this area from clotting. Okay, so we basically get this fluid pocket here that traps pathogens, and within that space is where we have antibodies and phagocytes and other defense cells that are going to neutralize those pathogens. And then finally down here we see chemotaxis is just the attraction of leukocytes or white blood cells to different chemicals that guide them to the site of injury. So I think when we talked about basophils, we talked about how they secrete different factors that attract neutrophils or other white blood cells that are really important, like eosinophils, to a site of infection. Okay, so that's how we contain and destroy pathogens. Oh no, this is the last part. It's tissue cleanup and repair. Okay, so here we have this really nice, swollen, painful foot and ankle. And so the first part of this that I want you guys to know is that monocytes basically turn into macrophages. Hopefully we know that by now, and then they'll just engulf and destroy damaged cells. So that's partially how we have some tissue cleanup. Edema or swelling, so we can see this ankle is very swollen, forces the valves of the lymphatic vessel or the lymphatic capillaries open, right? We saw this when we talked about the lymphatic system, but basically when we have edema, we get all of this fluid buildup in here, and so that really increases the pressure. When the pressure is greater out here than it is in here, what that's going to do is push on these valves and push fluid into the lymphatic vessels. All right, so that is what's going to promote drainage from the tissues. And so when we're draining lymph from these tissues, we're going to pick up fluid that contains bacteria, dead cells, debris, and all of those sort of things. So that's really big in cleaning up tissues. And if we remember the 4 cardinal signs of inflammation, one of them is swelling. So that's how you get your swelling. And another is pain. So pain's really important in the repair part of inflammation because it basically acts as an alarm signal that says to your body, hey, this is injured, you need to limit its use so that it can rest and heal. Okay, so pain is an important part of inflammation that helps with tissue repair for that reason. Okay, immune clearance is one of the 4 methods of pathogen destruction by the complement system. So let's go back to this slide here. We have this big cascade of reactions that is induced by all of these different proteins, and they result in 4 different things. Inflammation, which we just talked about. Now we're going to talk about the next 3, immune clearance, phagocytosis, and cytolysis. So immune clearance, basically what happens, and I couldn't find a good picture of this, so hopefully you can close your eyes and imagine what I'm talking about, but basically what happens is these proteins, these complement proteins, they're going to bind different antigen-antibody complexes to red blood cells. And you might be saying, well, why would it do that? Then it's going to mark the red blood cells for destruction. But what actually happens is those red blood cells arrive to the liver and the spleen, and there are macrophages inside the liver and the spleen that strip those complexes off the red blood cells, and then they're going to destroy them. Okay, so that's immune clearance. That's one of the effects of the complement system. Phagocytosis, we already know a lot about that. This is another one of the methods of destruction through that complement system. And essentially what happens is we have these complement proteins here, and they're going to bind to whatever the pathogen is. And in this picture diagram here, it's a bacteria. Okay, so the complement proteins are going to bind to the bacteria. They are here now, one molecule, are bound together. And what happens is this phagocytic cell over here has these receptors that are going to bind to the complement proteins. Now they're all bound together and they can phagocytize this pathogen more easily. Okay, so that's another effect of the complement system. And then finally, we have cytolysis, which is the final effect of the complement system that we're going to talk about. So, cytolysis occurs through this thing called the membrane attack complex. And what happens is these complement proteins basically bind to a pathogenic cell. So, let's say this is a bacterial cell. And so these complement proteins will bind to the cell and then they'll start binding to each other. And as a result, we get this ring called the membrane attack complex, given the name. And so what it does is sort of like the perforins that we talked about with the natural killer cells, it forms a hole in the target cell. Okay, so instead of releasing granzymes, it just basically creates this inability for the bacterial cell or pathogenic cell to maintain homeostasis because things are flowing in and out of it, and eventually it will die. And then the last part of innate immunity that we're going to talk about today is fever. Okay, so fever is an elevation of body temperature that results from trauma, infections, drug reactions, brain tumors, all sorts of different things. But it is the second protective process of the immune response. So first protective process we talked about was inflammation, and the second one is now fever. So it's often more beneficial for us to allow our fever to run its course. Most of the time, or I know for me, I start to get the chills and I'm like, oh God, I need some ibuprofen right away. But most of the time it actually is beneficial for us to let our fever run its course. And the reason for that is because it promotes the activity of these interferons. Okay, so let's go back and take a look at interferons so we can remember what they do. It's right here. Okay, so interferons, if you remember, are the proteins that are secreted by cells that are under attack by viruses, and they alert their neighbors. The other benefit, they also activate natural killer cells to come target them. Okay. Oopsie, I went a little too far there. So what happens is if we take some kind of fever-reducing agent, then it's not that our interferon cells won't work, it's just that they work better at a higher body temperature. And at a higher body temperature, we also inhibit the reproduction of bacteria and viruses, and also accelerate our tissue repair. So in those ways, it is more beneficial to let a fever run its course than to take medication to reduce it. On the flip side of that, excessively high fevers can be dangerous. So what happens is when we get a fever, sometimes it speeds up different enzymatic pathways and just sort of results in chaos and cellular dysfunction. And so sometimes we can get seizures, comas, or irreversible brain damage. Pyrogens are the last thing that we're going to talk about today. They're fever-producing agents. So different examples of pyrogens are, for example, chemicals on the surface of a bacteria or virus that are secreted, or other chemicals that are secreted by the macrophages or neutrophils when they're attacking these pathogens. Okay, so essentially what happens is these chemicals are picked up by your body, and then they travel to the hypothalamus to stimulate neurons there to raise your body temperature. And so as a result, you will start to shiver to generate heat, and also cutaneous blood vessels, so blood vessels that are right under the skin, are going to constrict so that we don't lose too much heat at the surface of the skin. So that's why you'll sometimes to the touch feel cold and clammy, but internally your temperature is actually running really high. Okay, so practice questions for this section. Give me some examples of the protective proteins that we talked about. We talked about interferons, so tell me what those do. We also talked about the complement system. What are the results of that big cascade of reactions? Give some examples of cells that are involved in the innate immune response. So we talked about leukocytes and macrophages. Tell me about, neutrophils, eosinophils, basophils. What do each of them do? Tell me about natural killer cells, all those sort of things. Give some examples of protective processes involved in the innate immune response. So that's going to be things like fever and inflammation. Tell me, what are the benefits of those? What are macrophages? So give me some examples of macrophages and tell me where they're found. How do interferons protect against disease? Remember, they alert their neighbors and do some other things, so tell me about that. How does the complement system protect against disease? So remember, and this is sort of a reiteration of this first question here, but tell me about what are the results of the complement system, and how do each of them function? And then what are the pros and cons of having a fever or the febrile response? And then finally, what are the cardinal signals of inflammation? There are 4 of them, and we talked about redness, heat, swelling, and one other. All right, so if you guys have any questions, you can shoot me an email and we can Zoom, or I'll answer your questions through email, whatever you want to do.